let's get back to the medicine real quick. We got a lot of science level folks on the call today who are interested in how the regulatory challenge is exacerbated by highly personalized therapeutics, where the therapeutic is perhaps specific to a certain mutation. Dr. Shams, how is that, in your opinion, how is that regulatory challenge kind of amplified?
Right, that's a great question. And over the years, we started off, my career started off in small molecules, and you just throw something at it and you live with the safety and see what happens, and then half the products don't make it because of safety issues. Then my next stage was in biologics. We got more precise. Okay, we are going to go after this one antigen, and we have this engineering capability, and outcomes became really, really fabulous, right? And then the next stage is even more precise now, upstream. And at this point, I'm looking at just 20 amino acids, or 20 on the nucleotides, and trying to be as specific and as precise as possible to deal with a certain condition. And so I've gone through the journey, and I love it because it gives you control over what you want to achieve, and the biology of the disease and the pathobiology have improved so much that our understanding has really, really emerged, and that is what is leading to this preciseness. However, as I've grown through the system, my view is precision is awesome, but personalization is more important.
Mm-hmm.
I think, and that poses its own regulatory conundrum as you personalize medicine, because each patient is different, and we are using techniques where we are doing population-based studies in a normal distribution of the curve, and then we are missing the extremes, and with precision we can address some of these issues, but the patient is not monolithic. It in many cases has other issues that have to be dealt with. And so for me, precision is awesome, but there is a lot of work to be done on personalizing care. And that's where the patient really will benefit in totality. So that's where I am philosophically speaking.
Yeah, when I turn this question over to Dr. Hunter, I have to tell you, Dr. Hunter, I am on this one in particular, I'm super torn as to whether to ask you to respond with your FDA hat on or your REGENXBIO hat on. I'll let you choose, because I'm super intrigued by either answer, but you can get where I'm going with that. What are your thoughts?
Yeah, if I could start with my FDA hat on for a second, just to say that, the regulators are thinking about this a lot. Just very recently, they were asking for feedback and engagement around this, right? And the stats are out there. There were twice as many drugs in the past 8 years than for rare diseases, and often referred to as orphan drug diseases, than in the previous 8. And for genetic diseases, with recent approaches to testing molecular diagnoses, as everyone knows, sort of being able to get to very rare genetic diseases and thinking about developing drugs tailored to the specific variants that may be possible for these patients is often referred to as N-of-1 therapies. And that's because sometimes they're designed for the population of maybe even one person. But with this brings a set of challenges and considerations that have not been seen before for typical drug development. So they've noted, the agency has noted that, disease is often rapidly progressing, requiring prompt medical intervention, and therefore the needs to proceed need to be very quick to have a chance at helping these individuals. And so they've asked for feedback, especially in the space of antisense oligonucleotides, where they say at this time, development of this type of product is most advanced, and really wanting to hear from, what does it mean for efficacy to be evaluated, and what are some, processes that make sense for regulatory submission to the FDA? And what about sort of institutional review board reviews? And what does it mean for informed consent when you're talking about one patient? So it's definitely something that is being discussed. It's been discussed for the last couple of years at the agency, but lately there's been a lot more efforts around moving this field forward and applying some of the real world considerations to that. I should also point out that Dr. Marks has been talking about the Bespoke Gene Therapy Consortium for a few years now, and this is a new sort of collaborative effort for the agency along with NCATS and industry to come together to really think about, specifically for AAV gene therapy, what can be done for some of these underserved, ultra-rare populations. And this is both around science and manufacturing, and really to demonstrate that it's doable, that even if there are challenges that with some pilot programs, perhaps, that the field can really set the example of how it can be done. And REGENXBIO is a member of this consortium, and we are looking forward to participating in this larger group to really make some advances in this space.
Great. We're running short on time here, so I'm gonna just ask a couple more quick questions of you both, and then we'll get things wrapped up. And I wanna start with you, Dr. Shams. Which of your previous, so you've had a lot of experience, as I've said, with big bio and into small bio. Which of those experiences with the agency? I mean, you've seen drug development through to commercialization. Which of your experiences are perhaps the most analogous to RNA or gene therapy? In other words, can any precedent sort of be drawn from your experiences with other therapies? And if so, what best practices can be inferred?
Yes. I think there is a lot of learning in the biologics space, generically speaking, that can be leveraged by everybody, including the agency, as we review newer sort of biologics, if you want to call them that, which is genes and RNA therapies and everything else. I think there are several overlaps that go across from the non-clinical side to the clinical side to the manufacturing side. That those learnings can be leveraged very effectively, I think. And so I think the space, at least in my case, I think what I learned with the development of a biologic at Genentech, for example, I have carried that over, not in one-on-one situations, but generally speaking, it serves you really well as you go into other areas that are similar, where the challenges may be similar. So that's been my experience, is that, So another reason that I just mentioned was the precision side of things. It is very precise. The immune response and the biologic side of things is very precise. And so it has, in my experience, again, the efficacy side of things looks a lot sort of better on the biologic side than on the small molecule side. That's my bias. But overall, I think that sort of carries over into the precisionness of the newer technologies that I'm working on now at ProQR with oligos and manipulation of the RNA. So I think there is a lot of lessons to be learned there and carried forward.
Yeah.
We don't have to reinvent the wheel. That's for sure.
That's a good thing. Yeah. Dr. Hunter, would you agree? You've been, you've seen the biologic side from the FDA, and obviously now at REGENX. Would you agree that there's some precedents out there that can serve as best practices for our audience?
Yeah, I agree. I think we can look back at antibodies and production innovation that really sparked what many call the inflection point in advance of those products. And the agency has, reflected back around sort of how it was widespread adoption of safe and effective platforms, and sort of how they thought about developing, for example, fully human monoclonal antibodies that weren't rejected by the immune system. So I think there's a lot there to look back on and pull out some lessons learned to apply going forward.
Great. Okay. Final question for each of you. And I'd like to wrap things up by asking our two. Here we go with another corny, another corny eye joke. I'd like to ask our two ophthalmic visionaries to gaze into their crystal balls and do some prognosticating. What does the future hold? Dr. Shams, let's start with you. Where's the regulation of gene and RNA therapies headed from here? What are your predictions?
My predictions are this is just the beginning, and it's full of hope and a lot of progress. And I think there's no way going back or stopping where you are. It is what it is, and both us as the innovators on the outside and our regulator colleagues and friends have no choice but to move forward. And this will lead to many, many good things, and not in the distant future, in the near future, I think. And by near, I mean 5 to 10 years. I'm not talking 30, 40 years.
Mm-hmm.
So I'm very, very hopeful that a lot of these technologies are going to bring in a lot of benefit to a lot of people.
Dr. Hunter, your thoughts?
Yeah, I totally agree. I think hopeful, I think great potential. It's going to require some patience, but it's exciting and really looking forward to being part of this and working with industry partners and with our collaborators at both the regulatory agencies and elsewhere to really move this field together.